Professor Carafit here to talk to you about the mitosis meiosis worksheet. I originally made a video where I drew all of this, but it looked like crap, so I thought I'd do this using images. Now, the worksheet starts. Now, first things first, it's very important you do the worksheet before you watch this video. The worksheet's useless if you watch this video before you do it or while you do it. Do it first. This is the only way you can catch your misconceptions and fix them. Okay, so first things first, do it. Do the worksheet, pause this video, go do it, then come back. Okay, all right, now you're done. All right, so the worksheet starts off with this image here, and this is a image of a cell in as if we could see the chromosomes in G1 phase. In reality, these chromosomes would be in the form of chromatin and they wouldn't be condensed, but this helps us to visualize what's happening. So we have chromosome pair 1 and chromosome pair 2. This cell is 2n equals 4. 4 chromosomes in the diploid cell. And this is chromosome pair 1, so these are homologous chromosomes. White chromosomes are maternal. Hatched chromosomes are paternal. You can see they have the same genes on them. They're just designated R and B in this case, gene R and gene B, and the numbers represent which alleles they have. So you might think of R as eye color, and there's blue and brown, and B might be hair color, and there's brown and blonde. So number 1 means brown, number 2 means blonde, or whatever. There's different alleles, different versions of those genes. And then there's chromosome pair 2. And also there's a gene there, gene D, and there's D1 and 2. The black boxes here, those are your centromeres, okay? They can be in the middle, but they can be towards an end of a chromosome as well. So these are the centromeres. So these are the unduplicated chromosomes consisting of a single chromatid. That's what we see here, okay? Now, the first question is draw these 4 chromosomes after they have replicated as they would appear on the equatorial plate of metaphase. Keep track of the shading. This is important. Keep track of the shading and where the alleles are. That's really important. Students skip that all the time, but I can't tell you know what you're doing unless you actually draw it all. Now, I'm not collecting this worksheet, but you have to prove it to yourself that you know it. The question is then, how many chromosomes are in the cell and how many chromatids are in the cell? So let's have a look at what this would look like. So remember, each of those chromosomes are going to duplicate, and they're going to line up on the metaphase plate of mitosis. And here's what it looks like, okay? Is this what you came up with? Hopefully. But here's chromosome pair 1. Now remember, this is a diagram, so these are, you know, if I was drawing this by hand, these would be drawn more like this, and the chromatids would be touching, right? And they would all be lined up on the metaphase plate. You know, like that. I'm drawing the chromatids as boxes here, so they're easy to tell, easy to see what's going on. They'd be connected at the centromeres, you know, like this, you know, lined up on the metaphase plate, single file row. The chromosomes down here are duplicates. I mean, the chromatids down here on the south side are duplicates of the ones on the north side, so they have the same alleles on them. As the one above. All right, they're duplicates, they're exact copies. That's mitosis. It says, how many chromosomes are in the cell? Well, these are duplicated chromosomes, so there's 1 chromosome, 2 chromosomes, 3 chromosomes, 4 chromosomes. So there are 4 chromosomes. How many chromatids? 1, 2, 3, 4, 5, 6, 7, 8. 8 chromatids, in the cell. All right. There you go. So that's how you draw this in mitosis. Now, the next thing for you to tackle is turn this into meiosis. How would the chromosomes potentially line up in meiosis 1? And show 2 ways they could line up. This is to test your understanding of synapsis, homologous chromosomes coming together, and your knowledge of independent assortment. Make sure you do that. If you haven't done it yet, pause this video. Do it now. Do it now. All right, let's have a look. How would this line up? Here's one way they could line up. They could line up like this. Now here, students get this wrong all the time, and it's important that you check to see if you got it wrong. Okay, so they're lined up on the metaphase plate. So if I was drawing this by hand, here's the metaphase plate. We have a pair of chromosomes up here, and we have, or I should say, we have a pair of chromatids up there and a duplicated chromosome. We have a pair down here, for example, and the same thing for the other chromosome, right? And they technically would be, if I wanted to draw this really accurately, remember the chromatids are you know, this, in reality I should draw them like this, where they're, you know, they're touching and the metaphase plate is right here. But just giving us a little distance to see what's going on. Okay, so here's the metaphase plate. Here's the duplicated chromosome. They're in synapsis. They're paired up. In reality, they're touching. But like I said, I have distance on the drawing so you can see what's happening. This is one way they could line up. Notice all the maternal chromosomes are on top. All the paternal chromosomes are on the bottom. What's another way they could line up? Remember, it's 50-50 chance whether each pair, which one's on top or which one's on bottom, whether it's paternal on top or maternal on top. 50-50 chance for every chromosome. So in a human being, that means you have 2 to the 23rd possibilities of different kinds of gametes because of this arrangement. Another way they could line up. Here's another way. Notice here we have on top a paternal set and a maternal set, and the same for the bottom. Now this has a big outcome on what kind of gametes are going to form at the end of all this. Just to give you a quick example, let's draw the 2 gametes that would come out of this top set and the 2 gametes that would come out of this bottom set. So these 2 chromosomes here are going to end up in a cell at the end of meiosis 1. These 2 chromosomes are going to end up in a cell at the end of meiosis 1. Then in meiosis 2, sister chromatids separate. So this chromatid and this chromatid are going to go into this cell. This chromatid and this chromatid will end up in this cell. And down here, we're going to have that combination. So in this case, you're going to end up with every one of those cells has a maternal chromosome 1 or 2 and a paternal chromosome 1 or 2. They have, if there's a maternal chromosome 1, then there's a paternal chromosome 2, and vice versa. So there's variation in those gametes. Whereas in the first example, all the gametes have either all maternal or all paternal chromosomes because of the way they lined up. If you don't believe me, draw it out, figure it out. What you need to do to be successful as a student is, if I say like, and they'll all end up in the same cell, and you're like, okay. But if you don't understand it, the only way to learn it is to get out a blank piece of paper and draw it, okay, and figure it out. All right, the final exercise is crossing over. And it says, consider only chromosome pair 1 in meiosis 1 and show how a crossover event would occur if the crossing over event occurred between the R and B genes. Okay, what the heck does that mean? Right between the R and B genes is where the chromosome breaks off. Everything from that break-off point to the tip of the chromosome is going to cross over. Okay, so what would that look like? Pause this if you haven't done it yet. Okay, you've done it? Great. Here's my example. Assume in this case the metaphase plate is here going up and down. We have a tetrad, and here we have a entirely maternal chromosome. Here's an entirely paternal chromosome. But these 2 chromosomes here have undergone crossing over. They are recombinant chromosomes. Notice from here to here is paternal, but from the rest of it is maternal. And on this one, from the bottom to the middle is paternal, but from the middle to the tip is maternal. And now we have new combinations of alleles here. We have an allele from mom and an allele from dad on the exact same chromosome. This is terribly important to understand. Okay? Now, the final question then is, what would the gametes be that arose from this question? And so you need to remember that this one's going to go into a gamete, this one's going to go into a gamete at the end of meiosis 2, right? And this one is going to go into a gamete, and that one's going to go into a gamete at the end of meiosis 2. Because in meiosis 1, this tetrad is going to separate. In meiosis II, sister chromatids are going to separate. And you're going to end up with gametes that have each of those different chromatids, which are now chromosomes, in them. After they go from sister chromatids to daughter chromosomes, and they end up in different cells. And the worksheet only tells you to pay attention to this chromosome for crossing over, but just to help you avoid confusion, bear in mind the other chromosome that we kind of ignored also is going to end up in these cells, and it will also have crossing over. And so you didn't have to do this for the worksheet, but just to show you what that might look like, here's an example of the final, one of the ways these could finally end up in gametes. There are other combinations thanks to independent assortment and things and where crossing over occurred, but this is just an example. So the final gametes, remember we had a cell that was 2n of 4. There were 4 chromosomes in that original cell. Our final gametes here are n of 2. There are 2 chromosomes in each of these cells. These are now ready to go. Maybe they're eggs, maybe they're sperm. And notice I showed some crossing over here in the smaller chromosome as well, just so you could get a feel for that. So variation arises thanks to both independent assortment and crossing over. Independent assortment will mix up the proportions of paternal and maternal chromosomes in the gametes, and crossing over will mix up actual pieces of chromosomes, paternal and maternal. On homologous chromosomes, those pieces can swap places. Okay? All right, well, I hope this was a challenging worksheet, and I hope that you learned something. And if you're confused, that means that you just need to work through it a few more times. Okay? All right, good luck, and I will see you in class.